Hello class, for this video we will talk about the respiratory system. The respiratory system is going to be involved in gas exchange, so making sure that we have a supply of air that gets in close proximity to the blood capillaries so that oxygen can load into the blood capillaries, carbon dioxide can unload. So that's the primary function. It has some other functions, but we'll talk more about those in a minute. So to get that job done, you have to have a way of getting air from outside of the body into the lungs. And then you have to have specialized membranes for that transfer, the exchange of gases to be able to occur. So the respiratory system is going to reside in the head, the neck, and the thoracic cavity. And it's going to consist of a respiratory tract, which is going to be involved in having a passageway for air to get into the lungs. And then we also have a point in which we have the gas exchange occurring. So the air is going to enter through the nose into the nasal cavity. The air is then going to flow into the pharynx, which is also our throat. From the pharynx, the air goes into the larynx, then into the trachea, and then the trachea goes down to the lungs. The trachea branches and we form a bronchial tree. And we have a number of different, we have primary, secondary, tertiary, terminal, respiratory bronchioles. We have a number of different structures that are involved in that bronchial tree. We can divide the respiratory system into an upper and lower respiratory tract. So the upper respiratory tract, so somebody talks about an upper respiratory tract infection, that's going to be from the nasal cavity to the larynx. Lower respiratory tract, that's going to be from the trachea through the bronchial tree all the way down to the alveoli. These alveoli are very tiny air sacs, and they are arranged in grape-like clusters, and this is where gas exchange is going to occur. So each lung is going to have about 150 million alveoli, and then those alveoli are surrounded by blood vessels for the gas exchange and also transport to and from the lungs. And then we also have a lot of elastic connective tissue because the lungs need to be able to expand and then recoil back to their original shape. So here we can see the upper and the lower respiratory tract, the upper being the nose, the nasal cavity, pharynx, larynx, and then the lower respiratory tract being the trachea, and then the trachea branches into the bronchial tree inside of the lungs, and then the bronchial tree is going to dead end at the alveoli, this grape-like cluster. Besides upper and lower, we can also divide the respiratory system into what's happening in the functions. We can have a conducting and a respiratory zone. Conducting zone is about air transport, so air traveling, going in and out. So if it's part of the respiratory tract that's just involved in air going in and out, so inspired, expired air, then that's conducting zone. That is going to go from the nose, nasal cavity, through the bronchioles. And then the respiratory zone, this is about gas exchange. And this, we're typically looking at the alveoli as the respiratory zone, although we do have some respiratory bronchioles that can undergo gas exchange also. So respiration is going to be our process that's going to make sure all the cells of the body get oxygen and that we also can remove carbon dioxide from the cells of the body. But there are 4 separate processes that are occurring for respiration to occur. One, you have to have air in and out of the lungs. So that's your pulmonary ventilation. Then at the lungs, we have to have gas exchange occurring. This is also an external gas exchange. So it's going to be movement of gases between the lungs and the blood, so between the alveoli and the blood. This is where we load oxygen, unload carbon dioxide. Then once we have the blood loaded with the oxygen, we need to transport that blood that's high in oxygen throughout the body to make sure that the cells of the tissue have access to that blood. And then we have tissue gas exchange or internal gas exchange. And then this is movement of the gases between the blood and the tissue. So unloading oxygen and loading up, picking up carbon dioxide. There are other functions to the respiratory system, being able to produce sounds, vocalization, speech. That's going to be involved with the voice box, the larynx. We also have detection of odors, the nasal cavity. It's something that would have been talked about with special senses with AMP1. We also have that respiratory pump that helps return venous blood back to the heart and also helps with movement of the lymph through the thoracic as well as the abdominal pelvic cavities. We also have a role in maintaining the acid-base homeostasis, and this is because of the impact carbon dioxide has when it interacts with water and it forms carbonic acid, which then dissociates into hydrogen ion and bicarbonates ion. And we'll talk more about the relevance of that. And it also has a role in the production of angiotensin II, which we've already talked about, and the role angiotensin II has in blood pressure and fluid homeostasis. So let's go through the different components of the respiratory tract. The nose and nasal cavity, look at that. First, this is going to be involved in warming and humidifying air that's coming in. So warming the air as well as adding water vapor to the air so that when you get to the lungs, the air that comes in isn't cold, isn't dry. If it's dry, then you would lose even more water to the air than what we already do lose to the air. We also have filtering out of any debris that may have been inhaled. We secrete antibacterial substances to try and prevent infections from occurring. Olfactory receptors, so that sense of olfaction that was part of the special senses in A&P1, that's all there. And then resonance of the voice, and this is why when you have a stuffed nose, it changes your voice because it's going to impact that. So from the nose, nasal cavity, air is going to flow into the pharynx, and we have three regions: the nasopharynx, oropharynx, and laryngopharynx. So the pharynx in general is a muscular tube, and it's going to connect the nasal cavity as well as the mouth to the trachea and or esophagus. So it's going to be this muscular tube for shunting food, liquids, air to the appropriate tube it's supposed to go into. With the nasopharynx, this is going to be just behind the nasal cavity, and we have pharyngeal tonsils that are located here, and any of the tonsils be involved in trapping pathogens. The oropharynx, that is posterior to the oral cavity, and this is where we're going to have food in passing as well as air is now going to be passing through here as well. That's going to extend from the uvula to the tip of the larynx, and the tip of the larynx is the epiglottis. We also have tonsils here, the palatine and lingual tonsils are located here. The laryngopharynx, this is going to extend from your hide bone, which is in your throat, to the esophagus, and it's going to convey food to the stomach and then air into the larynx. And the epiglottis is going to work to try and ensure that food and liquid do not enter into the larynx to get into the trachea and down to the lungs. So here we have the three regions of the pharynx in different colors. So here we would see all three, and then the first region, most superior, is the nasopharynx, the middle region behind the oral cavity, oropharynx, and then the region of the pharynx that's going to be shunting foods and liquids into the esophagus or air towards the larynx, that is the laryngopharynx. So from the pharynx with the respiratory system, then we go to the larynx. The larynx is going to be the connection between the pharynx and the trachea. It's going to work to keep foods and liquids out of the lower respiratory tract. And then we also have vocal cords here for sound production. There is a series of cartilage that are found here. We'll just focus on a few of them. With somebody's Adam's apple, which is more pronounced in men than women, that is due to thyroid cartilage of the larynx that's producing that structure in the throat region. And then epiglottis, this is going to be made up of elastic cartilage, acts as a flap, and then it's going to cover the opening. The opening is called the glottis, so it covers the opening of the glottis to the larynx to try and make sure that food and liquid will not enter into the larynx. Then we have a few other cartilage, but we're not worrying too much about those right now. So here we have pictures of the larynx. We have our hyoid bone, and then if we go inferior to the hyoid bone, the first set of cartilage that we see is the thyroid cartilage, and then we have this, it's called laryngeal prominence, that is your Adam's apple. And we can also see, if we look posterior view, we can see this flap of cartilage, that is the epiglottis. And if we look at it from a mid-sagittal section, so from a side view where we've sectioned through the larynx, we can also see that epiglottis, and that epiglottis is capable of closing off the glottis so food stuff will not get into the larynx. This image also shows the vestibular fold and vocal fold, that's involved in your sound production. And the vocal folds, those are your true vocal cords. They are going to be inferior to the vestibular folds or your false vocal cords, but it's the true vocal cords or vocal folds that are involved in the voice and sound production. The vestibular folds still have an important role. They're going to be helping to close off the glottis during swallowing. We also have vocal ligaments, and these are going to be at the core of the vocal folds. They're elastic bands. They'll appear white, and there are muscles in the larynx that can pull on cartilage and that can rotate inward and adduct the vocal folds, or they can rotate outward and abduct those vocal folds involved in sound production. For your loudness, that is determined by the force of air. So quieter, there's less air, a force of air going through. Louder sounds, there's more of a force of air going through. And then the pitch, that's going to be due to the tension of those vocal ligaments as well as speed of vibration. From the larynx, air is going to flow into the trachea. So this is going to deliver air to our lower structures of the respiratory tract. And it's about 10 to 12 centimeters long, it's inferior to the neck, and it's going to extend to the mediastinum region, and then it'll branch. And when it branches, we start forming the bronchial tree. This structure has C-shaped rings on it, and the rings are made up of hyaline cartilage, and they're C-shaped rings that help hold the trachea open. We don't want the trachea collapsing, because if it collapsed, air would not be able to pass freely through it, whereas the esophagus, which is posterior to the trachea and involved in movement of food stuff, that's a muscular tube that does collapse. The trachea is not going to collapse, it's going to stay open. If you put your hand in your throat, like gently put your hand to your throat and rub up and down, you can feel the rings, those C-shaped rings of hyaline cartilage in your throat that are keeping that trachea open. So here we have the larynx, and then air is going to pass from the larynx into the trachea, and that trachea has the C-shaped cartilaginous rings in them. The trachea is going to branch, and that first branch will be primary bronchi, and then they'll branch again, and that's going to be our bronchial tree being formed off of the trachea. So trachea leads air into the bronchi, and the first branch off trachea will be the primary bronchi, and we have a right and a left. So you have a branch that goes to the right side, the right lungs, and then a branch that goes to the left side, to the left. The primary bronchi, they branch, and when they branch, then you form secondary bronchi. And then the secondary bronchi, they branch, and you form tertiary bronchi. And then they'll continue to branch again as we form this bronchial tree. So you can remember it, the primary is 1, secondary is 2, tertiary is 3. With each branch, the bronchi are getting smaller and smaller and smaller. And as the branching is occurring, the shape of the cartilage changes. We go from a C shape to rings of cartilage. And that's just going to help keep the passageways open as we get smaller and smaller branches. We also have smooth muscle around the bronchi that we're seeing formed here. And that smooth muscle, we can have constriction and dilation. And if we have constriction, we're making the airways smaller. It's going to reduce airflow. And if we have dilation, the airways are larger. It's going to allow for more airflow to go through that particular bronchi. And here we see the trachea, and then the first branch off the trachea will be the primary bronchi, and then we have a branch off the primary that is going to be secondary, and we'll have that on each side. And then the branches off the secondary, those will be our tertiary, and then we keep having branches until we get all the way down to extremely small bronchi, and they'll be the terminal and respiratory bronchi that will lead to the alveoli. So the smallest of the bronchial tree, those are the bronchioles. These are only about 1 millimeter in diameter. They lack cartilage, but they still have smooth muscle rings around them, and the bronchi will end up forming about 65,000 terminal bronchioles, and those terminal bronchioles, they will give rise to the smallest of the bronchioles, the respiratory bronchioles. And those respiratory bronchioles, they do still have a thin band of smooth muscle around them. And those respiratory bronchioles can branch into alveolar ducts, and then those alveolar ducts will terminate in alveolar sacs, and those are grape-like clusters of alveoli. Here we have an image showing what happens when we get to the terminal bronchioles. So terminal bronchioles, air flowing through them. Terminal bronchioles can branch, and when those terminal bronchioles branch, we get the respiratory bronchiole. The respiratory bronchiole, they can branch, and when you get a branch off the respiratory bronchiole, then we have alveolar ducts. And the alveolar ducts are going to take the air into sacs, which are our alveolar sacs, and the alveolar sacs are the cluster of grapes, with each individual grape being alveoli. This image also shows where we end conducting zone and start respiratory zone. So everything through terminal bronchioles is conducting zone. It's just passing air. There's no gas exchange happening. Once we get to respiratory zone, so the respiratory bronchioles, the alveolar ducts, the alveolar sacs with the alveoli, that is where gas exchange can occur. And you can see the large amount of blood supply that is being supplied to the alveolar sac and the alveoli to facilitate that gas exchange. This is a nice image because it is showing you the passageway that air takes entering in and then working its way all the way to the alveolar sacs. So let's look a little bit at the alveoli and special cells that are there that are going to help with facilitating the function for gas exchange and then also defense. So these alveoli, they're going to have elastic fibers around them for distension, expanding, and recoiling. They also have smooth muscle, thin bands of smooth muscle around them. A lot of capillary beds, pulmonary capillaries are around them. And then we have 3 types of cells associated with the alveolar. There's type 1 alveolar cells, type 2 alveolar cells, and then alveolar macrophages, or sometimes called dust cells. Type 1 are our squamous cells. So the flattened type of epithelial cells. About 90% of the cells in the alveoli are going to be these simple squamous epithelial cells. So they allow for rapid diffusion of gases, which is really important to meet that function. The type 2 alveolar cells, these are cuboidal. So they're still epithelial cells, but they are cube-shaped instead of the flattened squamous cells. So only about 10% of the alveolar cells are going to be the type 2 cuboidal, but they're incredibly important because they secrete a chemical that eventually will become surfactant. So they have a precursor to this chemical called surfactant. And surfactant's really important because it reduces surface area. So you can think if you ever had a balloon and it was really humid or moisture inside the balloon, and you try to blow that balloon up, it's very, very hard to do so. It's hard to break those bonds between the water molecules. They want to keep the balloon collapsed. Your lungs are similar. So you have a lot of water molecules inside of your lungs, and that's going to want to cause the alveoli to collapse in. We do not want the alveoli collapse. We want to make sure the alveoli stay open so air can get in and out easily. Surfactant reduces the interaction between water molecules to be able to keep those alveoli open. And then the other type are alveolar macrophages. So macrophages, these are part of your immune system, and they can undergo phagocytosis, so they can clean up debris. They can migrate to the bronchioles, where they're then going to be swept up and eventually swallowed. So if you have mucus in the lungs with these macrophages in it, they can be moved up, coughing in, moved up, kind of backwards from the bronchial tree to the trachea, up to the larynx, and then if it's not spit out, it can be swallowed into the esophagus, get into the stomach, and then the pH, the acidic environment of the pH, will break the macrophages and anything that might still be inside of them down. There is a junction between the simple squamous epithelium of the alveolar and between the endothelium of the capillaries. And that junction is our respiratory membrane. So we have this membrane across which gas exchange will occur, and this is junction site between the alveoli, specifically the simple squamous epithelium, and the pulmonary capillary with their epithelial tissue as well, and that's called the endothelium. So there's 3 major components to this respiratory membrane. The type 1 alveolar cells, so the simple squamous cells in the alveoli, the basal lamina of those type 1 alveolar cells, and that fuses with the capillary basal lamina. So alveolar cells fuse basal lamina, and then the capillary endothelial cells. That forms our respiratory membrane. The respiratory membrane needs to be very thin and to facilitate gas exchange. And we also have enormous surface area in the lungs to allow for efficient gas exchange as well. So if we look at an alveoli, when we look inside the alveoli, we see all these simple squamous, these flattened cells. Those are our type 1 alveolar cells. We also see some cube-shaped cells. Those are our type 2 alveolar cells. They're going to end up ensuring we have surfactant to keep the alveoli open. And then we have these irregular shape. These are our alveolar macrophage. They're going to be there to engulf debris, foreign pathogens. And then if we look at the junction between alveoli and the capillary, we have our respiratory membrane that's shown here. So the respiratory membrane is going to consist of the type 1 alveolar cells, the basal lamina that's been fused of the alveoli with the basal lamina of the capillaries. And then we have our capillary endothelium cell. So very thin membrane for short distance for gas exchange to occur. So we look at the organs that are referred to as the lungs. We have lungs on the left and lungs on the right side of the body. The left lung has 2 lobes, and there is a fissure that separates those 2 lobes called left oblique fissure, and it separates the left lung into a superior lobe and an inferior lobe. The right side of the lungs has 3 lobes. So the heart is located just left of the body, so there's a little less room for lungs on the left side because you have the heart there. The right side, you're not sharing that space with the heart, so there's more room for the lungs, so you have 3 lobes. And the 3 lobes will be a right superior, a right middle, and a right inferior lobe. And they're separated by a horizontal and then a right oblique fissure. So here we have the lungs, and lungs on the left, lungs on the right. You can see the space that the heart is going to sit. With the left side, we have the superior lobe, and then we also have an inferior lobe, and then we have a fissure that separates superior from inferior lobe on the left, and that's the oblique because it's going at an angle. The right side, we have right superior, right middle, and right inferior. The fissure between superior and middle is horizontal. It's going more horizontally than at an angle, whereas the fissure between the middle and the inferior right lobe, that's at an angle, that is the oblique fissure. So the lungs have serous membrane around them, and the serous membrane around the lungs is called the pleura. The pleura, like other serous membranes, has 2 layers to it. The outermost layer is the parietal pleura, and then the innermost layer is the visceral pleura. Between those 2 is a potential space or a cavity called the pleural cavity, and that cavity is filled with fluid. It has the serous fluid, and that acts as a lubricant because our lungs are expanding and then recoiling, expanding and recoiling. So there's a lot of movement which would create friction, but we have that serous fluid, the pleural fluid, in the pleural cavity to reduce that friction. Some of the processes involved in respiration, like our ventilation and our gas exchange, they are actually physical processes. So we want to talk a little bit about some physics to be able to understand them, and we'll look at pulmonary ventilation first, which is that's movement of air in and out of the lungs. And the air that's moving in and out of the lungs is going to do so because of pressure gradients. So you have a difference in pressure from one area to another is going to cause gases, air to flow, just like we had pressure differences in the cardiovascular system that allowed blood to flow. We have pressure differences, pressure gradients in the respiratory system that are involved in movement of air. And the air, those gas molecules, move from an area of high pressure to one of low pressure. Just like with the heart, the blood flowed away from high pressure generated at the heart towards areas of lower pressure closer to the capillary beds. And we have a law, there's Boyle's law, that is going to help with this, and it says that at a constant temperature and a constant number of gas molecules, so you're keeping the temperature the same, you're keeping the amount of air the same, the pressure and volume of a gas are inversely related. So that means that as you change the size of the container, the pressure is going to change. So if you have a really large container with, say, 3 molecules of oxygen, it's going to have relatively lower pressure compared to a smaller container that has the same 3 molecules of oxygen. So just change the volume, make it larger or smaller, that's going to change the pressure. And that's relevant for us because the lungs change size. When you inhale, the lungs get large, and when you exhale, the recoil, the lungs get smaller. So we see changes in the pressure within the lungs as we change the size of the lungs, and that's what Boyle's law is referring to. And when we are looking at ventilation of the lungs, there are a couple different pressures that are important for us. One is the atmospheric pressure. So this is due to pull of gravity on air, which we don't necessarily think about that, but air does have a mass. It's made up of molecules, so nitrogen, oxygen, carbon dioxide, water vapor. So there actually is a mass to it. So gravity has an impact on it. And at sea level, the atmospheric pressure is 760 millimeters of mercury. So that is one factor that's going to be involved in ventilation. Another one is the intrapulmonary pressure. So intra within pulmonary lungs. So that's going to be air pressure inside of the alveoli. This will change as you have inspiration and expiration. So as you're inhaling and exhaling, you change the size of the alveoli, and that's going to end up changing the pressure within the alveoli. Then there is an intrapleural pressure, and this pressure is in the pleural cavity. So in the space between the parietal and the visceral pleura, in that space there is a pressure. Usually it's about 4 millimeters of mercury less than the intrapulmonary pressure. So it's a little bit less than what's going on inside of the lungs. So when the lungs are not actively expanding, inhaling, or getting smaller, recoiling, exhalation, then the intrapulmonary pressure, so the pressure in the alveoli and the pressure in the atmosphere, are going to be equal. That's going to change when we have inspiration and expiration. So when we are inspiring, when we are breathing in, our lung volume increases, the lungs expand. So if the size of the container is getting larger, then the pressure in there is going to decrease. So the lungs get bigger, the intrapulmonary pressure decreases, it goes below atmospheric pressure. So atmospheric pressure is now high, intrapulmonary pressure is low, so air goes from an area of high pressure to one of low, so air goes into the lungs. And then as we exhale, the lungs recoil, so they get smaller. As the size of the lungs get smaller due to recoil, the intrapulmonary pressure increases because they're getting smaller, so that pressure increases. It increases above atmospheric pressure. So now the lungs are high, the atmosphere is low, the air moves from an area of high to low, so it moves from the lungs out. So air moves out of the lungs purely because the lungs are recoiling and changing the pressure inside of the lungs, and then the air leaves the lungs. So this shows the changes in those pressures. You don't need to worry so much about memorizing the exact numbers here. Just make sure you understand the concept that as we inhale and the lungs get larger, that means the pressure, the intrapulmonary pressure, decreases. It becomes less than atmospheric pressure and air flows in. And then as we exhale, the lungs recoil, they get smaller. That means pressure increases in the lungs. Pressure is higher in the lungs than in the atmosphere, so the air goes from the lungs out into the atmosphere and we exhale. I did mention intrapleural pressure, and this becomes relevant if for some reason the intrapleural pressure increases to either atmospheric or above atmospheric pressure. And this can happen if you have some kind of an injury that punctures through the pleural cavity. And then you can have the pressure in the pleural cavity equalize with the atmospheric pressure. And when you have that, then that can cause the lungs to collapse. So what's involved in inspiration and expiration that's actually changing the size of the lungs? And we have muscles involved in that. There are inspiratory muscles. These are going to be skeletal muscles and they are going to increase the volume of the lungs by increasing the size of the thoracic cavity, because the pleura, the parietal pleura, is attaching the lungs to the walls of the thoracic cavity. So if you change the size of that thoracic cavity, because the thoracic cavity is attached to the lungs, you change the size of the lungs. And we have our main inspiratory muscles, the diaphragm muscle, so that's going to be between the thoracic and the abdominal pelvic cavities, and then the external intercostal muscles, so between the ribs. They're the primary ones for inspiration. Inspiration is active. We have our inspiratory muscles contracting, expanding the thoracic cavity, expanding the size of the lungs. And then expiration, normal expiration, is actually passive as the inspiratory muscles relax and recoil. Now you can do a forced expiration, which is going to involve muscles, but your normal expiration is just a passive process as muscles relax and the lungs recoil because of their elastic properties. And then this visual is going to show the connection between changing the size of the thoracic cavity changes the size of the lungs. If we increase the size of the lungs, then we have inhalation occurring. When we have our passive recoil and the thoracic cavity gets smaller, the lungs get smaller, then we have exhalation occurring. This chapter has animations like our previous ones. Just make sure you go back to the actual PowerPoint to click on the link to see the animations. Here's another animation that you can view. So what other things can impact pulmonary ventilation besides our inspiratory and expiratory muscles? So we have airway resistance. So anything that's going to impede airflow, that's going to impact your ability to get air in and out of the lungs. Your alveolar surface tension, so surfactant should be there to keep the alveoli open, but if something happens where you're not producing surfactant, or preborn babies oftentimes do not have surfactant production occurring at a level to keep the alveoli open, and that then is going to impede ventilation. And then pulmonary compliance, which is going to be the ability of the lungs to stretch. So if we decrease compliance, then we have an impedance to ventilation. So we can use the resistance as a way to control airflow into the lungs, because we can have bronchial dilation and bronchial constriction. Bronchial dilation, so this is like vasodilation, the smooth muscles, and this time they're associated around the bronchi, they relax. And if they relax, that's going to increase the diameter of the bronchioles. That's going to allow for more air to enter into the lungs. The sympathetic nervous system is going to be involved in bronchial dilation due to norepinephrine's impact. And sympathetic, remember, that's fight or flight. If you're in fight or flight, you need to have a significant amount of oxygen to be used as an energy source, because we need oxygen for ATP production. So exercising, stress, that fight or flight, emergency, fight or flight, that's all going to cause bronchial dilation through stimulation of the sympathetic nervous system. And then we have bronchioconstriction. Here the smooth muscles are going to contract, decrease the diameter of the bronchioles. That would increase resistance. That would then decrease flow to the lungs. This can be triggered by inhalants. So you might have some inhalants that are irritating the system, and they would activate the parasympathetic neurons, or we might have activation for bronchioconstriction through inflammatory mediators. Bacterial, viral diseases can cause this. Influenza can cause this, because all of these things are going to cause symptoms that are going to increase the resistance in the airway. So here we can see our change in size of the diameter airway. So here it's open, smooth muscles relax. That would be the bronchial dilation. Here it's smaller, smooth muscle is contracted. This is going to be our bronchial constriction. This will decrease flow because we have increased resistance. This will decrease resistance, which means we increase the flow of air. So let's talk a little bit more about the alveolar surface tension. And we talked some already about surfactant, but we want to talk a little bit more about this. So if you remember from Properties of Water from 168, water molecules will interact with other water molecules because of hydrogen bonds. These H bonds or hydrogen bonds cause the oxygen of one water molecule to be attracted to the hydrogen of another water molecule. So the water molecules aren't independent, they actually have this desire connection to link up with one another, and that's due to formation of the hydrogen bonds. This creates a surface tension, just pulling the water molecules together, and can then pull the alveoli, the walls of the alveoli, close together too when we're looking at moisture in the alveoli. So in order to prevent or reduce that alveolar surface tension, we have surfactant. Again, it's produced by the type 2 alveolar cells. One end of the surfactant is going to be polar, which means it can interact with water molecules, and then the other end is going to be nonpolar, which means it doesn't like water molecules, it repels them. So surfactant's hanging out with water molecules, but then also has an end that keeps it from binding to other water molecules. And that effectively reduces surface tension. It interrupts the ability of the water molecules to form those hydrogen bonds with one another, and that's going to allow the alveoli to remain open. This scenario, we have surfactant present, and the surfactant, you can see these larger circles here. We have water molecule and then surfactant. Water molecule and then surfactant. So the surfactant is separating the water molecules, decreasing the surface tension, keeping the alveoli open. This scenario, we do not have the surfactant, so the water molecules are forming hydrogen bonds with one another, and it's not just on the outside of the alveoli. The water molecules from one side want to interact with molecules from the other side, and that pulls the alveoli in and collapses the alveoli. Here's another animation that covers lung compliance and airway resistance. We have a tendency to want to measure things, to use them as a guide for whether or not there's any kind of abnormalities in the system. And when it comes to pulmonary functionality, we use a spirometer. So it's just going to be a tool that's going to measure the volume of air that a person exchanges with each breath. So the volume of air coming in, or the volume of air being exhaled, and or the volume of air that might be being retained. So we have a number of pulmonary volumes that the spirometer can measure. So here's a table that has the different pulmonary volumes and capacities that can be measured in order to determine if there's any abnormalities with an individual in terms of their pulmonary volumes and their pulmonary capacities. And this graph shows all the different types of pulmonary volumes and how they relate to one another. And if you want some more information on that, you can look at the animation in the PowerPoint. So we want to start being able to understand gas exchange. But in order to understand gas exchange, we need to go back and talk a little bit about some physics again. And there's another law, so it's Dalton's law of partial pressures. And what it says is if you have a mixture of gases, which is what the atmosphere is, it's oxygen, it's carbon dioxide, it's nitrogen. So in the atmosphere, in the air, we have nitrogen, oxygen, carbon dioxide. That is a mixture of gases. So it's saying that the pressure, so the air pressure, is due to the partial pressure exerted by each of those individual gases. So nitrogen is going to be part of that total atmospheric pressure. Oxygen is going to be part of total atmospheric pressure. Carbon dioxide is part of total atmospheric pressure. So each of the gases making that mixture will be part of the pressure, and that is referred to as the partial pressure. And if we add all the partial pressures up, that gives us our atmospheric pressure. And the partial pressures are due to the percent that a gas makes in the mixture. So the partial pressure that would be the highest would be nitrogen, because that's the highest percent gas in the air. And then the lowest one would be the carbon dioxide, because that's the lowest of the percent gases in the air. So what we need to do is to figure out our partial pressures is to figure out the percent of the total that each gas exerts. So nitrogen is 78%, so if we take 0.78 times 760, that gives us the amount of the total, the amount of the 760 millimeters of mercury that is due to nitrogen. We can do the same thing with oxygen, do the same thing with carbon dioxide. So oxygen is going to be about 160 millimeters of mercury. So your individual gases all have a part or a role to play in the total atmospheric pressure. So when we want to look at gas exchange, we need to look at the partial pressure of oxygen and the partial pressure of carbon dioxide, because the same principles apply in terms of gases move from an area of high pressure to low. So oxygen is going to move from an area of a high partial pressure of oxygen to one of low partial pressure of oxygen. Besides the partial pressures in Boyle's law, we also have Henry's law. Henry's law is referring to the solubility of a gas. We care about the solubility of gases because we were looking at being able to move things like oxygen and carbon dioxide into and out of the blood. So nitrogen has a high partial pressure, but we don't see very much in the plasma because it's not very soluble. It doesn't want to go into a liquid solution. Oxygen has a lower partial pressure than the nitrogen, but we'll see some in the plasma because it has higher solubility. And then carbon dioxide has very low partial pressure, but it dissolves readily in the plasma because it has a higher, about 20 times more solubility than oxygen has in water. So the gases we see in the blood is going to be due to partial pressure properties and then also due to the solubility of that specific gas. So let's put these things together and look at pulmonary gas exchange, or our external respiration. So the gas exchange that's occurring between the alveoli and the blood capillaries in the lung. And here we will load oxygen into the blood, so from the alveoli into the blood, and then we will unload the carbon dioxide from the blood into the alveoli. So at the lungs, inside of the alveoli, we have about 104 millimeters of mercury for our partial pressure of oxygen, which means that the partial pressure of oxygen is pretty high in the lungs, and in the capillary, the partial pressure in the blood is only about 40 millimeters of mercury. So high in the alveoli, lower in the capillaries, so oxygen moves from the alveoli into the blood, and we load the oxygen into the blood. The carbon dioxide inside of the alveoli, it's only about 40, so it's relatively low in the carbon dioxide. Partial pressure carbon dioxide is low in the alveoli, but it's higher in the capillary. So the carbon dioxide is going to unload and move from the blood into the alveoli so it can be released from the body. If we look at the tissue level, when we look then at internal gas exchange, we see the opposite. So now we have cells of the tissue, so maybe a liver cell or a skeletal muscle cell, and then we are looking at a systemic capillary. We're no longer in the lungs. In the systemic capillary, we have a relatively high amount of oxygen, it's about 100 millimeters of mercury inside of the blood. Is relatively low in the cells. So the oxygen gets unloaded, moves from the blood into the cells, and it's then going to be needed by the mitochondria for ATP production. And we see the opposite with carbon dioxide. Carbon dioxide in the tissue is 45 millimeters of mercury, so it's higher in the tissue than it is in the blood, and then the carbon dioxide leaves the tissue and goes into the blood. So we are loading carbon dioxide at the tissue level. We load carbon dioxide into the blood, and we unload oxygen from the blood into the tissue. So it's moving into the tissues, into the cells, unloading it from the blood into the tissues and the cells. There is an animation that covers the pulmonary gas exchange. Its link is in the PowerPoint. The system is set up to have efficient gas exchange at the lungs to ensure that we have appropriate levels of oxygen and carbon dioxide. If we have too low a level of oxygen, that is called hypoxia. If we have too high a level of carbon dioxide, that is hypercapnia. So these two factors, too low of oxygen or too high of carbon dioxide, are monitored, and they can have significant impact on ventilation rates, which we'll talk about in a minute. And when we think about the lungs, we might think that all areas of the lungs are going to get the same amount of air, and then all areas of the lungs will have the same amount of blood flow going to them. But that's not the way it works. There is ventilation, perfusion, matching, or coupling. So ventilation is referring to the amount of air going to any given area of the lungs or any group of alveoli. Perfusion is referring to the amount of blood flow going out to any specific capillary bed in the lungs. And we match things up. So if we change the ventilation, then that's going to lead to changes in the blood perfusion. So if one area of the lungs is getting a large amount of oxygen and other areas not, then the area getting the alveolar ventilation is also going to get the blood supply to that. And then the opposite can happen too, that if we change perfusion, we change where blood is flowing to an area, then we change the ventilation. So the area that's getting a large amount of blood in the capillaries is going to get a large amount of air going to that specific alveoli group that is being served by that capillary bed. And it's the process of bronchial constriction and bronchial dilation that's going to regulate where the air is flowing to, so where you're getting your ventilation, and then vasoconstriction and vasodilation is going to regulate where the blood flows, where you're getting the perfusion. So this is summarizing that. If we have normal partial pressure of oxygen and carbon dioxide, then we're going to have perfusion occurring, normal perfusion occurring. If we have low oxygen in the alveoli, then we are going to have reduced perfusion occurring. If we have high oxygen in those alveoli, then we increase perfusion, have more blood going to that. And now if we look at the blood in the arterioles, so looking at the perfusion, if we have low carbon dioxide levels, then we're going to have essentially a low blood flow, so lower perfusion. Then we are going to constrict the bronchioles to decrease air going to that area. And then if we have the opposite, we have high partial pressure carbon dioxide in a given capillary bed, so large volume of blood, high carbon dioxide, then we are going to have bronchial dilation to ensure that oxygen air is getting to that alveoli for gas exchange to occur. Here's another animation you can view. Another animation, this one on tissue gas exchange. So gas exchange is occurring. We've talked about loading and unloading oxygen. Where is the oxygen going? And we've already talked a little bit about this because we talked about hemoglobin inside of the erythrocytes when we talked about blood, and that the hemoglobin binds to oxygen. So we want to look at how is this happening and what's going to influence whether oxygen is being bound to hemoglobin or whether oxygen is being released from hemoglobin. And just as a review, remember the hemoglobin is found inside of the red blood cells. We have 4 polypeptide chains. Each chain has a heme group, and each heme group has iron in it, and the oxygen is going to bind to the iron in the heme group. We load oxygen in the alveoli, and that's going to form oxyhemoglobin. And then when we unload oxygen at the tissue level, then we have deoxyhemoglobin. So in the lungs, we have our alveolus, and we have oxygen molecules inside of the lungs, and we have a higher partial pressure of oxygen in the alveoli than we do in the blood. So the oxygen is going to move from the alveoli into the blood, and it's specifically going to bind to the hemoglobin molecules inside of the red blood cells. And then when we are at the tissue level in the systemic capillaries, we have high partial pressure of oxygen, high partial pressure of oxygen in the blood, lower partial pressure of oxygen in the tissue. So the hemoglobin lets go of the oxygen, and the oxygen then diffuses into the cells. Another animation that you can watch in the PowerPoint. So we want to be able to load the oxygen onto the hemoglobin and then also be able to unload it. So there are factors that influence how tightly the hemoglobin is going to bind to oxygen, because we want to be able to load it and unload it. We don't want hemoglobin so attached, so bound that it doesn't let go of the oxygen. And the degree with which hemoglobin binds oxygen is called affinity or bond strength. There are a few factors that can impact the affinity. One is the partial pressure of oxygen. So if we increase the partial pressure of oxygen, we see more loading occurring. And if we decrease the partial pressure of oxygen, we see more unloading occurring. So there's this relationship that we can end up putting on a graph and getting an oxygen hemoglobin dissociation curve. So it's going to be the relationship between the partial pressure of oxygen and what percent of the hemoglobin is saturated, or what percent of the hemoglobin has oxygen bound to it. Here is an oxygen hemoglobin dissociation curve. And this curve is going to have the percent oxygen saturation, so going from 0% to 100% saturation. And then it also has the partial pressure of oxygen, so going from 0 partial pressure of oxygen up to 100. And we see that this curve is roughly S-shaped. And as we increase the partial pressure, so as we were going increase in partial pressure, we see an increase in the percent saturation. So the higher the partial pressure of oxygen, the higher the saturation. And then if we go in the opposite direction, as we lower partial pressure of oxygen, we see lower saturation of the hemoglobin with oxygen. So this supports what we were talking about. We have high oxygen levels in the lungs, the partial pressure of oxygen high in the lungs. So that's going to cause hemoglobin to have higher affinity and grab hold of the oxygen, which is what we want to happen in the lungs. And then when we get to the tissue level, we have a lower partial pressure of oxygen at the tissue level, and that's going to cause hemoglobin to let go of the oxygen, which again is what we want to happen so that we can deliver oxygen to the cells. Another video in the PowerPoint you can watch. There are factors besides the partial pressure of oxygen that will influence that affinity or attraction between hemoglobin and oxygen. And these are temperature, hydrogen ion concentration, partial pressure of carbon dioxide. So if we increase any of those, the temperature increases, we have a higher concentration of the hydrogen ions, or we increase our partial pressure of carbon dioxide, that will decrease hemoglobin's attraction to hemoglobin's attraction to oxygen, and that will allow oxygen to be unloaded. And then also there's a molecule BPG, which is 2,3-bisphosphoglycerate, that will also cause unloading of oxygen from hemoglobin. It reduces the affinity if that level increases. So where would we expect to see an increase in temperature, increase in hydrogen ion concentration, and an increase in partial pressure of carbon dioxide? That is what we see naturally at the tissues. So the tissues are going to have a little slightly higher temperature, more hydrogen ion concentration, and more carbon dioxide, partial pressure carbon dioxide at the tissue level. So when we are at the tissue levels, we actually see a shift where we are unloading oxygen because of these factors, as well as the fact that there's also a lower partial pressure of oxygen at the tissue levels. So this image is showing that relationship between hemoglobin and temperature, hydrogen ion concentration, and partial pressure of carbon dioxide, and whether we are loading or unloading oxygen. So normal temperature, hydrogen ion concentration, partial pressure carbon dioxide, we're going to have some unloading of the oxygen happening. If we have an increase in any of these, so temperature increases, and this might be due to exercise, then we see there's even more oxygen being unloaded. Or if the hydrogen ion concentration increases, so we see more hydrogen ion concentrations, then we get even more oxygen being unloaded. Or if we see an increase in the partial pressure of carbon dioxide, then we get even more oxygen unloaded than normal. And this causes a shift. So if the dotted line, this dotted line is our our normal, dotted line is our normal scenario, then if we have the increase graph plotted, this is our solid line, this is going to be the change in the hemoglobin oxygen dissociation curve, the oxygen hemoglobin dissociation curve, due to these changes. And this is a shift to the right. It's called a right shift curve. And this right shift curve is favoring unloading of the oxygen to facilitate the needs of the cells. So we should have an understanding now of how oxygen is transported, with primarily it being bound to hemoglobin. There's a little bit of oxygen that is dissolved in the plasma, but it's a very small amount. The bulk of oxygen is bound to hemoglobin. Now, carbon dioxide is also transported in the blood, but it's a little bit different than what we see with oxygen. We actually have 3 mechanisms that are all significant to help transport carbon dioxide. Some of the carbon dioxide is just dissolved in plasma. About 7 to 10% of carbon dioxide is transported this way. Some of the carbon dioxide is bound to hemoglobin. It's about 20%, and this is forming carbaminohemoglobin. And then the bulk of the carbon dioxide is inside of a bicarbonate ion, so about 70% is going to travel in the bicarbonate ion. So where does the bicarbonate ion come from? Carbon dioxide, when it interacts with water, we have an enzyme, carbonic anhydrase, that's going to facilitate this. It forms carbonic acid, and then carbonic acid dissociates into the hydrogen ion plus the bicarbonate ion. So the bicarbonate ion, so here's carbon dioxide, that bicarbonate ion ends up inside, or here's carbon dioxide ends up inside of the bicarbonate ion, and then it's transported that way. This also explains why we see an increase in the hydrogen ion when we have an increase in carbon dioxide levels. So if the partial pressure of carbon dioxide increases, that increases hydrogen ions, which would make the area more acidic or decrease the pH. Here's another animation that you can watch. So here's the formula I just had up. So carbon dioxide is going to be inside of the bicarbonate ion. Carbon dioxide formed by the tissues interacts with water in the plasma. We form carbonic acid. The carbonic anhydrase, that's an enzyme that's going to act with the carbon dioxide in the water to form carbonic acid. And carbonic acid naturally dissociates into bicarbonate ion, where the carbon dioxide is stored, and then the hydrogen ion. This reaction can move in the forward direction, which is what's going to happen at the tissues where we have a large amount of carbon dioxide, high partial pressure carbon dioxide. And then it can go in this reverse reaction, which is what's going to happen at the lungs when we want to take the carbon dioxide that is inside of this bicarbonate ion and free it back up to be able to release carbon dioxide at the lungs. So what's happening here? Carbon dioxide is going to diffuse into the erythrocytes. That carbon dioxide is going to be converted into carbonic acid because of the carbonic anhydrase. Carbonic acid dissociates into the bicarbonate ion and the hydrogen ions. Those hydrogen ions will stay inside of the red blood cell because they bind to hemoglobin, but the bicarbonate ions, they're going to leave and they will enter into the plasma. And when they leave, when bicarbonate ions leave, chloride ions will enter. So carbon dioxide from the tissue has been formed due to aerobic respiration. So the aerobic cellular respiration, and one of the byproducts of breaking down something like glucose is we form carbon dioxide. So that carbon dioxide leaves the tissue, goes into the blood, gets into the red blood cells. Carbon dioxide and the water interact, form carbonic acid. That dissociates into the hydrogen ion and bicarbonate ion. Hydrogen ion is going to bind to hemoglobin, and the bicarbonate ion is going to leave the red blood cell to get into the plasma, and when it does so, we exchange bicarbonate ion for chloride ion. So we have a chloride shift. And when we get to the lungs, we get to the pulmonary capillaries, we need to see oxygen or see carbon dioxide reformed. So the carbonic acid is going to be reformed first inside of the erythrocyte. So the hydrogen ion is going to let go of the hemoglobin. The bicarbonate ion is going to re-enter into the erythrocyte. So we have our raw materials to build carbonic acid. So the carbonic acid is going to be formed. That then breaks down into water and carbon dioxide, and carbonic anhydrase is going to be involved, catalyzing this again. And once we have our carbon dioxide reformed, it can diffuse into the alveoli to be released from the body. So now we are at the lungs. So here's our alveoli. We have our bicarbonate ion that was in the plasma is now going to enter into the red blood cell, and we have that chloride shift again. So bicarbonate ion enters, chloride leaves. The hemoglobin's going to let go of its hydrogen ion, so we have hydrogen ion and bicarbonate ion, so we can reform the carbonic acid. That breaks down into water and carbon dioxide diffuses from the blood into the alveoli so it can be released from the body. So this system, this carbonic acid bicarbonate ion system, this works to transport carbon dioxide, but it also works as a buffering system. So a buffer system is going to be anything that's going to try and reduce pH changes. So try to keep the pH in a fairly normal range. And in the blood, the pH stays within a range of about 7.35 to 7.45 on a pH scale. So if we want to buffer changes in pH, we need to be able to have a weak acid and a weak base to be able to either absorb hydrogen ions to prevent it from becoming acidic, or to be able to release hydrogen ions to prevent it from becoming too basic. So we have a way of increasing the hydrogen ions in the solution. So if this reaction moves forward, we are increasing hydrogen ions. So if something is too basic, we can counter that by moving in this forward direction and increasing the hydrogen ion concentration to make it more acidic. If something is too acidic, then we can move this reaction in this reverse direction and take the hydrogen ions out, decrease the hydrogen ions in solution by moving in this reverse direction. And if something is too acidic, then we move in the reverse direction to start to make it more basic. So if you have changes occurring where the blood is becoming a little bit too acidic or a little bit too basic, we can alter the direction that this reaction is occurring in to intentionally impact the amount of hydrogen ion in solution to either make it more or less acidic based on whatever way the pH is changing. So because carbon dioxide levels influence pH through changing the hydrogen ion level concentration, then our ventilation rate can influence the pH of the blood. Because if we increase ventilation, then we get rid of more carbon dioxide that can make it more basic. If we decrease ventilation, then we're holding on to more carbon dioxide that can make it more acidic as we increase the number of hydrogen ions. So changes in ventilation can influence blood pH as we alter the partial pressure of carbon dioxide, the amounts of carbon dioxide. So if we are hyperventilating, we have a very rapid rate or depth of breathing, that can allow us to get rid of more carbon dioxide. So we're expiring more carbon dioxide. This is going to decrease the partial pressure of carbon dioxide in the blood, which is going to increase the pH. It's going to make it more basic, and that causes a respiratory alkalosis. And some natural things that can cause this: fever, anxiety, head trauma. Those are natural things that can cause that respiratory alkalosis. And one of the things you do for someone who's hyperventilating is you put a paper bag over their mouth so they breathe in the air they just exhaled. Because if you are too low in carbon dioxide, you need to increase carbon dioxide levels. Breathing in air that's high in carbon dioxide, so the air you just exhaled, is a way to counter hyperventilation, bring the partial pressure of carbon dioxide back to normal, and then that can counter that increase in ventilation that's been occurring due to hyperventilation. Now, we can also have an impact of hypoventilation, not having enough of a ventilation rate or depth of ventilation, and that would cause you to retain carbon dioxide, and that would cause you to increase the partial pressure of carbon dioxide, makes the blood more acidic, so you decrease the pH, making it more acidic, and that is the respiratory acidosis. And pulmonary neurological conditions can cause that. Your PowerPoint does have this big picture of respiration visual, so it's really good visual to evaluate how well you're understanding these concepts. Can you explain this? Do you know why one thing is leading to the next? If you have any questions on that, let me know. I'll help you with it, but it's a good tool to be able to evaluate how well you're understanding what we have covered. So let's look at the control of ventilation. So your normal ventilation is called eupenia, and this is going to be controlled by groups of neurons in the brainstem. So we have the pons and we have the medulla. The medulla is the primary structure involved in with the ventilation control and is going to act as a pacemaker. But the pons also has to play a role. The pons is going to be involved in trying to have smooth transitions between inhalation and exhalation because it can regulate the depth and rate of breathing that's set by the medulla. In the medulla, we have two regions. We have a ventral respiratory group, and then we have a dorsal respiratory group. The dorsal respiratory group, that is getting signals that are coming in from around the lungs, from the aortic arch, so signals are coming in, and then that dorsal group is going to be integrating stretch receptors and the chemoreceptors. And then the ventral respiratory group, that's going to be sending signals out to our inspiratory muscles for our rhythmical breathing. So for control of ventilation, we have to be monitoring what's happening in the body, and we have chemoreceptors in two different places. We have central chemoreceptors and then we have peripheral chemoreceptors. And those chemoreceptors, the central ones, those are going to be scattered throughout the brain. So medulla, midbrain, hypothalamus, cerebellum, we have chemoreceptors there. The chemoreceptors here are primarily looking at partial pressure, carbon dioxide, and pH of the blood. Because of the connection with partial pressure of carbon dioxide levels, we see changes of pH. So increasing the partial pressure of carbon dioxide will decrease pH. Decrease in partial pressure of carbon dioxide will increase pH. And because ventilation rate is tied to partial pressure of carbon dioxide, it also gives us an indirect measure of the need or not need for oxygen. Like, do we need to have more ventilation or not? So if our partial pressure of carbon dioxide get too high, then that's indirectly telling us that we need to have increased ventilation to ensure we're getting enough oxygen. So we are monitoring the pH levels of the extracellular fluid within the brain and also within the cerebral spinal fluid. And if we have high partial pressures of carbon dioxide or an acidic environment, that will trigger a hyperventilation increase in ventilation rate. If we have low partial pressures of carbon dioxide or we have basic conditions, then that would trigger hypoventilation or decrease the ventilation rate. Here, if we have high partial pressure of carbon dioxide, high number of hydrogen ions, the body's going to detect that with our central chemoreceptors, and then we are going to get a change such that we are going to increase the ventilation rate to be able to get rid of excess carbon dioxide. That will take us back to our normal levels. If we have too low partial pressure of carbon dioxide or too low hydrogen ion concentrations, again, central chemoreceptors are going to detect this. They will then send a signal that is going to trigger a decrease, a hypoventilation, a decrease in the ventilation rate. So you end up retaining carbon dioxide levels so we can return back to our normal level. We also have our peripheral chemoreceptors. The peripheral chemoreceptors are going to be located in the carotid arteries in the aorta. In the carotid arteries, we have the carotid bodies. In the aorta, we have the aortic bodies. And these are going to be highly sensitive to partial pressure of oxygen. So if we're changing the partial pressure of oxygen, these cells here will be detecting that and then causing a change in ventilation rate. So if we have a decrease in the partial pressure of oxygen, we get below 70 millimeters of mercury, then triggering carotid bodies will result in an increase in the rates and the depth of ventilation to be able to restore the partial pressure of oxygen back to normal levels. So this table summarizing that information, looking at the partial pressure of carbon dioxide and hydrogen ion concentration, that's primarily going to be our central chemoreceptors. Looking at changes in partial pressure of oxygen, that's going to primarily be our peripheral chemoreceptors. And then we also are looking at changes in stretch of the trachea and the bronchi. These signals are all sent to corresponding areas in the pons or in the medulla to be able to get the appropriate response. So we might see hyper or hypoventilation depending upon what's happening with partial pressures of carbon dioxide and oxygen, as well as hydrogen ion concentrations. So this is our topic on the respiratory system. Let me know if you have any questions.